Imagine you're getting older. You've spent a lifetime in the sun, and one morning you notice a little red bump on your forehead or your arm. It doesn't hurt, but it's growing fast. On the surface, it appears so innocent that it's often brushed off as a simple cyst or an annoying bug bite. But in just a matter of weeks, that spot becomes a large, dark purple mass that crusts and bleeds. These growths are the result of an exceptionally rare and highly aggressive skin cancer called Merkel cell carcinoma, which often strikes when a person's immune system is weakened. Because this cancer mimics harmless skin conditions so effectively, a physical check in the doctor's office can't tell us enough. To identify it and save a life, we have to look much deeper. Right now, we need to go past the surface and look inside the skin layers to see what's actually happening. Panels B and C show what the pathologist sees under a microscope. The cancer cells are packed together, and inside their centers, you can see a gritty, speckled pattern that scientists call salt and pepper chromatin. Finding these speckled cells creates a major problem for the doctor. Because of their generic shape, they form a vague category called small, round, blue cell tumors. Without further testing, these cells look exactly like lung cancer that has spread to the skin. Since the shapes alone don't tell the whole story, pathologists use specialized chemical markers to make the cells reveal their identity. This process is called immunohistochemistry. You can think of it as using highly specific chemicals that act like glowing paint. Doctors use chemical tags designed to latch on to specific proteins inside the cells. When they find an exact match, they lock on and light up. To find our specific cancer, scientists look for a protein marker called CK20. Look closely at panel C. In the lab, a positive result shows up as a distinct pattern of glowing dots clustered right next to the center of each cell. This distinct dot pattern provides the proof needed to distinguish skin cancer from lung cancer, allowing for a final diagnosis of Merkel cell carcinoma. Now that we've identified the cancer, how did these normal skin cells go rogue? In 80% of cases, the trigger is a biological hacker called the Merkel cell polyomavirus. This virus splices itself into the host cell's DNA. Once inside, it takes over the cell's biological machinery and forces it to divide endlessly. The remaining 20% of cases are caused by a different threat: severe ultraviolet radiation from the sun. Decades of harsh sun exposure physically batter the cell's genes. Eventually, they accumulate so many errors that the cell's internal safety switches break. Whether caused by a virus or radiation, the resulting tumor requires a highly advanced treatment strategy. Once the cancer is identified, doctors can bypass standard chemotherapy and use advanced immunotherapy. This treatment takes the molecular brakes off the patient's own immune system, allowing the body's natural defense cells to recognize and destroy the tumor. The success of this approach is remarkable. Compare the bleeding mass seen earlier with this patient's forehead, showing a completely healed surface after treatment. But there is a remaining paradox. Even though we call it Merkel cell carcinoma, scientists now believe the cancer doesn't actually come from Merkel cells, because normal Merkel cells aren't even capable of dividing. Instead, researchers found a strange behavior. The virus intentionally breaks its own protein, the large T antigen, which acts like a molecular key. That broken piece wedges into the cell's growth control machinery, permanently jamming it in the on position. We have the tools to track this cancer and the weapons to defeat it. Yet, exactly which type of normal skin cell it hijacked remains an unsolved mystery. If you enjoyed this deep dive into skin cancer, please like, subscribe, and comment below. This video was brought to you by Synesthetic Systems LLC.